Okay, thank you everybody for joining us today in the WormBase webinar. Today I'm going to talk to you about our author first pass pipeline and one of the great resources we have at WormBase, Texpresso Central. So first, just a quick overview of today's talk. So I'll start with author first pass, basically explain what author first pass is. I'll have a really brief vocabulary lesson. I'll give you a glimpse behind the scenes about how Author First Pass works, explain to you how you can find out about whether or not your paper has been processed for Author First Pass, and then we'll go through a demonstration of the Author First Pass form with a real paper. And in the second half of the talk, I'll talk about Texpresso Central, explaining what Texpresso Central is, how you can access it, another quick glimpse behind the scenes to explain what Texpresso Central does. And then we'll also have another live demo of how to search the C. elegans literature using Texpresso Central. So what is Author First Pass, or as we sometimes refer to it at WormBase as AFP? So Author First Pass is really a way for the C. elegans community to help alert WormBase curators to the information that is in your papers. So you've been working really hard in the lab and you've written up your results, your paper's been accepted and published, and you'd like to get your results into WormBase as quickly as possible, and we would too. So there are some key pieces of information that will help us do that. And that is, what entities did you study in your paper and what data types? So just for some quick AFP vocabulary, and this will help as I go through and explain how the form for Author First Pass works. So what do we mean by entities? So entities are basically physical things is a way to think about it. So in WormBase, we think of these as things like strains, genes, variations, transgenes. And so it's the kind of thing you could actually hold in your hand, a plate with worms of a particular strain that have a variation in a specific gene and maybe a rescuing transgene, right? So these are what we think of as entities at WormBase. Data types, on the other hand, are really the kinds of experiments that you did. So maybe you looked at phenotypes produced by RNAi or as a result of genomic variations. Maybe you've looked at anatomical expression patterns or studied site of action. Maybe you did some co-IPs or yeast-2 hybrid to look at physical interactions, or you're using C. elegans as a disease model. These are all the kinds of things we think of at WormBase as data types. So a little glimpse behind the scenes of what happens for Author First Pass. So like I said, once your paper gets published, we're alerted to its publication by a daily query that we do at WormBase for PubMed for the keyword elegans. So once the publisher has submitted your paper to PubMed, we'll be alerted to that. We then manually download a copy of the paper, usually a PDF. That paper gets processed in a PDF to text conversion. And then we use our pattern matching and machine learning applications to look for, like I said, the kinds of entities that you've described in your paper and the kind of data types or experiments that you've done. That process also identifies the corresponding author in the paper. We'll match that to an email address that we can extract. And then we will actually email that address with a form for you to fill out the Author First Pass, or a link to the form to fill out our Author First Pass information. So every Wednesday, we process new papers that we have and we send out email messages. So look in your inbox if you've recently published a paper, and there will be a message from the WormBase AFP team that will ask you to help WormBase curate your paper. And in that email message, like I said, there will be a link to the AFP form where you can look at the entities and data types we've extracted and verify those for us. Okay, so at this point, I'm going to actually go to our author first pass form and show you what this looks like and how this works. So for this demonstration, I'll actually use a paper that was recently published in the Sternberg lab in the journal Genetics. So when you click on the link in your email, this is where you will arrive. And this is the overview page for Author First Pass. So a couple of key features that I'll point out. So the information in the Author First Pass form is organized according to data type, and there are a number of different widgets here on the left-hand side for each different type of information. Now, as you go through the form, we would recommend that you systematically look at the information in each of these widgets, as sometimes our machine learning methods don't always get things exactly right. And any help and feedback that you can give us to verify data types will help improve our methods and make the process more efficient for everyone. There's also a link in the form to our set of FAQs that can help guide you in how to fill out this form, and release notes as we continue to make enhancements to our software and our methods, and we release new versions. Currently, we're on release 2.0 for Author First Pass. So what I'll do is I'll just use two of these widgets as an example to show you what kind of information we're looking for and how you can fill out the form. So let's start with the overview, which is where we have the information about genes and species. So if you're going to fill out the form, you got the link, first check up on top to make sure that we have the correct WormBase person for you. If you want to send your email to maybe one of the other authors on the paper to fill that out, that's fine. You can do that. They'll just need to change their user so that we can track who actually filled out the form. So if we need to get in touch with you for any reason to clarify things, we can do that. If someone doesn't have a WormBase person ID, there's a link out to the form to WormBase that you can actually request one. So I'll illustrate what we have in the form with respect to entities by using the genes here. So our methods will go through and scan the full text of papers for every mention of a WormBase gene that we know about. So one of the main tasks of WormBase is to keep as up-to-date entity lists as possible. So we'll use, say, the list of all the genes in WormBase to see what we can identify in the paper, and then we'll list them here. And we list both the name and the WormBase gene ID, so you can always verify the gene in WormBase if you want to. So what we're looking for with entities like genes and strains, variations, are those things that have really been experimentally studied in the paper. We know sometimes that people may use other genes in their studies, say as genetic markers, or maybe for co-injection, but those aren't really the things that we want to associate with the paper. We really want to associate things like genes with a paper where there's something about the biology of that gene with respect to C. elegans that have been studied. So what we ask authors to do is to look over the list of genes that we've identified. If we've missed any, you can add from the WormBase gene list, and this is an autocomplete list. If there's any that we've gotten wrong, you can select them, say for example, and remove them from the list. Also, if there's a particularly long list of genes here, you can filter that list to just look at a subset. So once you've verified the experimentally relevant entities, there are other gene features about genes and species in this overview page. If you want to request a new gene name, you can do that. This will take you out to a WormBase data type form where you can request a new gene name from Tim Shadle, who manages our C. elegans nomenclature, although we strongly encourage people to have pre-approved gene names before you publish. There's also a section here if you want to report a new gene sequence connection, you can do that. Or if there's a gene model correction or update for WormBase, you can flag that here and add free text details. Likewise, we present the list of species that we think are experimentally studied in the paper. And if any of these are not correct, you can remove them or you can add new ones from the WormBase species list. So at the end of every widget, once you've finished verifying the data, just click on Save and Continue, and that will automatically take you to the next widget in the AFP form. So what I will do at this point is I'll skip through some of these widgets and I'll just show you our data type verification using the phenotypes and function widget. So we have several different machine learning methods that use the full text of papers to determine whether or not there are experiments about different types of phenotypes, for example, allele phenotypes, RNAi phenotypes, or transgene overexpression phenotypes. And wherever you see this TPC powered, that means that that's a data type that we have a machine learning method for. When it's not there, it means we don't have one yet, but we're still actually very interested in getting author feedback on whether or not you think the paper has this kind of data. So for this particular paper, we were able to identify a transgene overexpression phenotype, and that's correct for this paper. But our machine learning methods actually missed the allele phenotype. So if that was the case for your paper, just click on allele phenotype, and that will let us know that this was actually a false negative. And this will alert the WormBase curators that there is actually allele phenotype data in your paper. And one of the other nice things about phenotypes in WormBase is that we have a separate community curation pipeline for curating phenotypes. And so we have a link out where you can add details in a structured way in our phenotype curation form. And then that will then go to WormBase curators for verification. And for functional data, we have flags for things like enzymatic activity or other gene function. If you have information for these, you can just click on the box and type in some free text details. So you might say in this paper, AFL2 is a member of a family of transcriptional elongation regulators, something like that. And that will go to the WormBase curators to give us a heads up of what kind of information is in the paper. Again, when you're all done with this widget, click Save and Continue, and that will take you to the next one. So at the very end of the process is our last page, which is Comments and Submit. So here what we can do is you can update your contact info, which WormBase keeps, so that if we ever need to get in touch with you for your AFP or to let you know about the next webinar, you can do that here. We keep track of the intellectual lineage at WormBase. You can update that from here as well. Importantly, if you have any unpublished data from these studies or any data that you think might make a great brief publication, you can get in touch with our micropublication team to publish in that journal. And then lastly, if there's anything about your paper that you think we really should know, if we missed, or if you have any comments or feedback about the process, we'd love to hear from you. And so you can add those with free text here. And then very importantly, when you're all done, click on Finish and Submit, and that will let us know that this paper has been completed for author first pass. Okay. So just a few new things to mention. So we have been working with our web team to acknowledge community curators on WormBase paper and person pages. So coming soon, there will be a new widget on the WormBase person page for community curation, and that will acknowledge and indicate any of the papers that our community has curated for author first pass as well as for phenotype. And then in the publication section, there will also be a flag that this paper has been community curated. So we really appreciate the efforts of our C. elegans community, and we want to make it more public to acknowledge everyone who has helped us with our community curation pipelines. Okay, so lastly, just to quickly summarize again the author first pass process. So by participating in author first pass, you will help us, one, validate a list of entities that are mentioned and experimentally studied in your paper, and help us flag papers for specific data types for curation. Any of the submitted data is included in WormBase. We try to do this as quickly as possible. This happens on a weekly basis. So we process papers and send out new emails every Wednesday. So if you get one of those emails, follow the link there and fill out the AFP form. So if you have any questions or comments along the way, feel free to contact us at help.afp@wormbase.org. Okay, so now we'll go to the second half of the talk, which is about Texpresso Central. So Texpresso Central is a great tool that we have at WormBase, and it's available to everyone. The URL is here, celegans.texpressocentral.org, and it allows users to do full text search of the C. elegans literature. So the Texpresso Central bibliography currently contains over 27,000 full text papers that we have at WormBase. And it allows you to do searches throughout that full text on both a document and sentence level. And so I'll demonstrate some of those searches in a little bit. But first, let me review some of the key features of Texpresso Central. So like I've said, it has the full text of the papers that we have in the C. elegans bibliography. You can perform simple keyword searches on the Texpresso corpus. You can also combine those with Boolean operators. You can perform category searches. So categories are either groups of related terms or entity lists. And that can help for sort of performing kind of broad searches. You can search on either a document level, or you can perform sentence level searches. Sentence level searches are particularly good for finding particular facts or experimental findings in the literature. All the search results from a Texpresso Central search are returned as individual matching sentences. So you actually see the full sentence that match your search criteria. And you can, for either open access papers or papers for which you have a subscription, view those matching sentences in the context of the full text. Okay, so a little bit again behind the scenes as to how Texpresso Central works. So basically what Texpresso Central does is indexes the full text of papers. So similarly to the author first pass, it will take PDFs or PMC open access papers and convert those to text. It then indexes the full text for keyword searches. And then additionally, it actually classifies the words and phrases in the full text according to, say, WormBase entity lists, ontologies that are used for WormBase curation, and some custom categories that we've created at WormBase that we use for curation that would also be very helpful, though, for the community to find specific pieces of information. So here are some examples of the kinds of categories that we have in WormBase. In Texpresso Central, things like gene lists, lists of variations. There's a category for genetic interaction terms. And an example of an ontology category that we have would say be the disease ontology. So I will now go over to Texpresso Central and we'll give you a demonstration of how this works. So this is the Texpresso Central homepage. And so just a couple of key features. So you can always find the number of papers that are in the current corpus. There's a link out to a published paper about Texpresso Central. Now you can perform a keyword search from either the search boxes here on the home page, or what I typically do is go out to the advanced search page, which you can access up here from the menu, because that will give you many more options for searching the C. elegans literature. So a couple things to point out here on the search page. Like I said, you can search through the document, or you can search on a sentence level. You can enter keywords here. They can be case sensitive, and you can also conduct Boolean searches from here. If you want, you can add one or more of the Texpresso categories to your search, and I'll demonstrate how to do that in a bit. And there are also additional search filters here that you might want to use. So you can exclude keywords, or you can filter your results according to specific authors, journals, publication years, accessions like PMID, or type of paper. Say you only want to look at review articles. One thing that I often use particularly is the sort by year function, as that will just float the most recent papers up to the top of your returned papers. Okay, so I'll just do a quick demonstration here. So I'm just going to search for the FA4 gene and say that I'm interested in finding all the papers that mention FA4. So I'll type that in and perform the search. And in about 1 second, Texpresso returns matches in 720 documents. So because it's looking over the full text, Texpresso is really, really good at finding mentions of things in the C. elegans literature. If you were to say go to PubMed and search on FA4 and elegans, you'll only get about 87 papers returned. So this means that Texpresso is really powerful at finding things in the C. elegans literature, but it also means that sometimes if you're looking for specific pieces of information, you might want to do more targeted searches. So let's say I'm actually really interested in FA4 as a transcription factor and what its targets might be. And I want to see if I can find papers that will give me some clue as to its targets. So if I was going to do a search like that, I would actually now do a sentence search. And since I'm interested in FA4 as a protein, I'm going to capitalize, do a search for capitalized FA4, make that case sensitive. Since I'm looking for targets, I'm going to add a keyword bind to see if I can find instances where people have described binding to specific promoters. And I'm actually interested in target genes. So now what I'm going to do is I'll actually add the category for the list of C. elegans genes. And so right now where that list lives is here under WormBase curation. If you scroll down and open up the menus, there's a category of genes and proteins. And I'm going to specifically select C. elegans genes. So then it will confirm your category selection here, and I'll just go ahead and click OK. And then I'll perform this search. And so now what we've done is this has narrowed my search down considerably, right? And I've only got 15 documents and 20 sentences to look at. So let's take a look at the search results now and see what Texpresso Central can do. So for every search, that you perform, you'll see the list of matching documents here. And so it will always present the accession, which is usually, but not always, a PubMed identifier, the paper title, the journal the paper was published in, the publication date, the type of paper, which is typically either a journal article or a review, and the relevancy score, which defaults to showing you the most relevant papers on the top. So if you want to explore a little bit more about your matches, you can click on this arrow, open up the resulting paper, and what you'll get is some additional bibliographic information about your matched paper. If you want, you can go to the abstract and read that. But then this will show you actually the kinds of sentences that were returned from the query. So this says further chromatin immunoprecipitation, ChIP qPCR analysis revealed that FA4 had the ability to bind to the promoter regions of these 3 genes and refers to a supplementary figure. So what you're seeing is each of the criteria that I gave for the search: capitalized FA4, the term bind, and the list of C. elegans genes. And so this is a great way to combine keyword and category searches with Texpresso to find specific pieces of information in the C. elegans literature. And if you want, you can click to view these sentences in the context of the full text. Okay, so that is our demonstration of Texpresso. A couple of other features that you might be interested in. The browsers menu actually enables you to browse and search the categories. So if you want to learn a little bit more about what categories we have in Texpresso, you can do that. If you want, you can also create a Texpresso account, and you can upload some of your own papers and make a custom corpus and those papers can be processed and made searchable by Texpresso. Okay, and so lastly, before we get to question and answer, I just want to acknowledge everyone who's part of the WormBase and Texpresso teams. This is very much a group effort, and so we really appreciate everyone's contributions. Particularly though, I want to acknowledge everyone who's involved in the Texpresso and author first pass pipelines. Valerio and Daniela are key in developing AFP. Michael and Valerio have developed Texpresso Central. Juan Carlos helps with all of our curation pipelines and tools. Cecilia, many of you may have heard of. She is the curator for all of our person information in WormBase. And Daniel Wang ensures that we have PDFs to process for AFP and Texpresso. And then lastly, special thanks to Sybil, who does our web development and has helped us work on the WormBase person pages where we can acknowledge everyone's community curation efforts. Okay, so thanks everybody very much for your attention, and we can go ahead and take any questions.
Thank you, Kimberly. That was wonderful. I hope everyone enjoyed Kimberly's presentation. So if you're interested to learn more, we published a paper on the AFP, and we published a paper on the Texpresso project. I will post the links in the chat in a minute. So we already have a few questions in the chat, especially from Alejandra. Thanks, Alejandra, for asking all these questions. The first question Alejandra has is, can you also fill the AFP form if you are the first author? The rationale is that first authors would usually not get the email for not being a corresponding author. And the comment is that often corresponding authors are too busy and may ignore that email, and then the information doesn't get on WormBase. Do you want to take this one, Kimberly?
I can, but if you want, you can go ahead.
Okay, so basically anybody with the link can access the form and fill it out. So in case the author, the first author does not get the email, you will need to ask your PI to forward you the link. So currently, we basically send the link to the form to the first email address that is linked to the existing person ID in WormBase that we find in the full text of the paper. So please go contact your PI, tell the PI to be on the lookout for the AFP emails, and it would be a great opportunity for you to become a community curator, which is linked to your other question. If anyone can become community curators, what you need to do to become one. Basically, any author on the publication can be a community curator. You don't have to do anything specific. We welcome your contribution. I hope, Alejandra, that answers your question, and you can answer in the chat. The second question is, how long does it take in average from publication until the paper is included in WormBase and the AFP form email sent out to the corresponding author? Kimberly, do you want to get this?
Sure. So that's a great question. So like I said, for WormBase, our primary, if not exclusive source at this point of being alerted to publications is PubMed. So as soon as a paper is in PubMed, we'll get the alert that it's there. And then if there is a more or less final PDF available, meaning that it's not still in proof, then we can get that paper and process that for the AFP. And then as soon as we've processed it, like I said, we have a weekly pipeline that will send emails out. We're hoping to improve the speed at which we do that. But if you have recently published a paper and you haven't gotten an AFP email from us, feel free to get in touch and we can see if we can get the paper processed for you as quickly as possible.
Thank you, Kimberly. The next question is if AFP and Texpresso also process supplementary data and text, or only the main paper. And maybe Valerio is the best person. Valerio is the AFP developer. Valerio?
Sure. So we're talking about Texpresso here, right? Texpresso includes supplementary material if it's in PDF form. So everything that it's a document as a supplementary material would be included and merged into the main paper for Texpresso. We currently not include images and videos or anything from which we can't extract text from the supplementary material. But when you do a search automatically in Texpresso, you're matching the main text as well as the supplementary material.
Great. Thanks, Valerio. So just to finish up the AFP part, Alejandra is asking who she can contact if she wants to flag a paper she published a couple of months ago, but is not on WormBase yet. You can write an email to help at AFP. Well, I need to look at the correct email now, but you can contact the AFP team.
help.afp@wormbase.org. Right?
Thank you very much, Chris. Thank you. Okay, great. Moving on, Mark has a question. He says he's confused. What did adding the C. elegans gene category does to refine this search further? Kimberly, this is related to Texpresso.
Texpresso search, sure. So let's go back to the example and sort of the question that I was asking with that search. So I was actually interested in knowing what C. elegans genes might be a target for the FOB4 transcription factor, right? So I could just do the search by maybe just looking for capitalized mentions of FOB4, right? but I wanted to specifically find targets. So I looked for capitalized FA4, the protein, and the term bind, right? But then I really wanted to hone in on those sentences that specifically mentioned a C. elegans gene, right? So that's why I included that category. So then if I'm doing a sentence scope search, the results have to be all the sentences that mention FA4, the protein, and have the word bind, and mention a C. elegans gene. So it's a really specific targeted search. But by including the C. elegans gene category, I made sure that the sentences that I got back mentioned a C. elegans gene. And does that answer your question?
Mark says, yes, that makes sense. Thank you. Thank you, Kimberly. Another question is how do papers get included in Texpresso Central? Kimberly, you want to take this one?
Sure. So as long as we have a PDF or PMC open access version of the paper, it will be included in Texpresso Central. We try at WormBase to bring in every paper that's published that uses C. elegans in some way experimentally. And so, right, as long as we have a copy of the full text, it will get included in Texpresso Central. We're trying to do updates, I think now roughly every 2 to 3 weeks. Michael can correct me if I'm wrong. We might be able to do it even more frequently. But if there are papers that you might expect to be in there that you're not seeing, let us know. And then we can take a look at that to see either if we're missing something, or if for some technical reason we're not able to include it in Texpresso.
Great, thank you, Kimberly. We have other 2 questions related to Texpresso from Laura. Is it possible to exclude the references of a paper from the search? I can imagine citations including your search terms when citing paper didn't. Valerio?
Yeah, so at the moment, we are searching also in the references because we don't have a module to do sectioning of the papers, but we're currently working on it. So it's a future feature that Texpresso will have to be able to select sub sections of the papers and do the search only on those. Not at the moment, though.
Great, thank you, Valerio. And Mark has another quick question. He has been using www.texpresso.org/celegans other than the celegans.texpressocentral.org that we posted. He's asking if there is a big difference between the two. I'll redirect this question to Michael Muller, who should be in the audience, who has just updated the site.
Sure. So the celegans, texpresso.org/celegans is going to go away. This is an old system, old software. The website looks different, and there are not as many features as in the new system. So we're actually going to retire that website pretty soon this year. So from now on, only celegans.texpressocentral.org will be the search engine to go to.
Thank you, Michael. Mark, did that answer your question? You can answer in the chat. It's been a very active Q&A session. Thank you for all the questions. While we wait for more questions, I'd like to start a poll, and we would like to ask a few minutes of your time to answer the questions. I'm gonna let it run for 2-3 minutes. Can everyone see the poll? So Laura says that she can see the poll, but it won't let her submit. Laura, you can answer in the chat. Did you arrive at the end of the poll? You answered all the questions? Just click other and answer number 2. Okay. Thank you.
So Daniela, there's a couple other questions. We missed one of Alejandro's, and I think Stavros has a related
Yeah, we'll
question.
let the people finish voting. They are seeing question 6 out of 7. So we're waiting still 10, 15 seconds. And then I'm gonna end the poll. So maybe because the question is from Stavros to Michael, he said Stavros asks he cannot sort the columns in search results table or dump it to a text file. Is that correct? And Michael confirms it's correct. So I'm gonna end the poll. And I can share the results. So we have 5 people that did not previously participate. So please do participate, and in author first pass, get in touch anytime with us if you have questions. Get in touch with your advisor if you haven't received emails yourself. And a couple of people said that they did not publish suitable papers. A person did not have time, which is very reasonable. Some have not been asked. Again, if you're not being asked and you published, get in touch with us and we will allow you to submit your data. The person that used the form says it was easy to use. We are glad to hear that. But again, send us any feedback if things are confusing or if you need us to modify the documentation and things are not very, very clear. And then if the form covered all the data described in your paper, yes, and not available, obviously, for all the people that have not participated. And we're glad to hear that some people already know that helping WormBase curate your papers will make your research more visible. So thanks again for answering to the poll. Okay, I'm going back to the chat questions. I see Alejandra has another question for Michael. It would be nice if it would be possible to download results in CSV in the future. In this way, data could be processed further. Michael, is that duly noted?
He responded. He said he'd put it on the
Okay,
to-do list.
responded.
Yeah, that's a very good point. I actually would like that as well. And I think
Okay.
Laura says the same.
Great, great. Thank you, Chris. I have too many windows open here. Okay, well, I think that we have addressed all your questions, hopefully. Thanks again, Kimberly, for a wonderful webinar. And thanks everyone for attending today. Thank you for the great questions. We hope you found this informative, and please put our webinar series on your calendar. The link to the schedule and sign up will be posted again in the chat. I'm gonna do that right now. And stay safe, and we hope you can join us next time. Thank you very much.
Okay, thanks, everybody.
Thanks.
Thank you.